Hello, students. This is my first try at doing a lecture on screencast, so hopefully everything works out all right. I'm missing you guys. Hopefully we'll see each other again soon, but I know you guys are all doing well with everything, and hopefully you're getting your academics completed and taking care of business. So let's go ahead and get started with Chapter 14, Section 2, Human Genetic Disorders. And our key questions for this section are: how do small changes in DNA molecules affect human traits? And what are the effects of errors in meiosis? Maybe you can hear my dog barking behind me. It's kind of annoying. But first, let's move. You can see the highlights. So if you would like to highlight also, the highlights are there for you. I don't know how you highlight it, but mine are there. How do small changes in DNA molecules affect human traits? Well, small changes can have large effects. We talked about mutations earlier in our sections. We know about the different, whether it's a point mutation, frame shift mutations, some of the chromosomal mutations like deletions and duplications. But just small changes can affect the amino acid sequence, like we saw the frame shift mutation, and that can directly affect the phenotype of a person, which would be the physical traits of that person. Some examples of disorders that are caused by individual genes. One would be sickle cell disease, or sickle cell anemia, as it's also known. There is one polypeptide in hemoglobin that is defective, and it will cause a misshapen red blood cell. And that misshapen red blood cell is sickle-shaped, which means it looks kind of like a crescent moon. And as they travel through the bloodstream, they don't carry oxygen very well, and they can also get stuck in the capillaries, which are very small blood vessels that connect the arteries and veins. And if they get stuck, you could have what are called sickle cell episodes, and that can damage whatever tissues or cells or organs that this episode is occurring. So that is one example of a disorder caused by an individual gene. Another example of a disorder caused by an individual gene is cystic fibrosis, or CF. Cystic fibrosis it's common in people of European ancestry. I can tell you we have had teachers at school that have had cystic fibrosis, and there are people in our community that have cystic fibrosis. It is, once again, caused by a small gene mutation, a deletion of 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator, or CFTR for short. But really, all you guys need to know is that while it's a recessive disease, so you need 2 copies of the disorder for it to happen. People with 1 copy will have some normal function, which they're usually just considered a carrier of it. But if they have the 2 recessive alleles, then they would have cystic fibrosis. And you can see some of the problems. There's your amino acid that is being deleted. You can see that the function of the cell membrane is not working properly. And the main problems would be clogged lungs, clogged membranes. There's a thick mucus there that causes clogging of the lungs and breathing passageways. And there's certain therapies that people with cystic fibrosis have to perform daily in order to clean up the lungs, and some of them involve patting and shaking the body to get them to clear the mucus so they can breathe. So there's going to be breathing problems. There's also going to be problems with intestines and digestion, also with some mucus in the intestines. Another disease that is caused by a small problem would be Huntington's disease. It is caused by CAG codon repeating, and some symptoms would be deterioration, uncontrollable movements. The problem here is it does not occur until middle age. It is a dominant disorder, but people don't know it until they're in their 40s. And if it is a dominant disorder, there's a 50-50 chance they can pass it on to their kids, which is kind of sad for them because they've probably already had kids if they reach middle age, so they would not have known if they were, if they had the problem or not. There are tests now that you can find out if you have Huntington's disease, and then you would have to make the decision on whether to have those tests or not to find out. Let's look at a video of Huntington's disease now, if I can find it. Here it is, so we'll go ahead and listen to this.
Hi, my name is Lauren Byrne, and I'm a research fellow at UCL Huntington's Disease Centre. My research career in Huntington's disease began in 2014, but in reality I've been researching Huntington's disease for the most part of my life. I grew up in a family affected by Huntington's disease back in Northern Ireland, and experienced it firsthand with my aunt and uncle who had it whenever I was a child. I've only memories of them being in wheelchairs. I started to notice the subtle signs in my dad whenever I was a teenager, and every year I've watched him deteriorate mentally and physically ever since.
I was naturally drawn to studying Huntington's, but what really fascinated me about the disease was that despite researchers knowing the exact cause of Huntington's disease since 1993, there still remains so much unknown about this extremely complex brain disease. Huntington's disease is a genetic, neurodegenerative, and ultimately fatal disease with devastating impact on individuals and entire families. Huntington's causes problems with a person's ability to think, behave, and move. Over time, the disease causes progressive functional decline, leading to a loss of independence and the ability to perform daily activities. Huntington's typically strikes a person in the prime of their life, between the ages of 30 and 50, although symptoms may appear earlier or later. Today, clinical diagnosis is made when movement problems are obvious. However, subtle changes known as prodromal or premotor onset symptoms often appear years earlier. Each child of a parent with Huntington's has a 50% chance of inheriting the disease, which affects men and women equally. We call this pattern of inheritance autosomal dominant. About 1 in 10,000 people live with Huntington's today, with many more at risk, including over 30 individuals within my own family. Everyone has 2 copies of the Huntington gene, which contains a section of repeated genetic letters known as CAG repeats. The Huntington protein is produced throughout the body, and although the normal function is not yet fully understood, it is thought to be involved in many biological functions, including the development of the nervous system. Huntington's disease occurs when one copy of the Huntington gene has a CAG repeat expansion over a certain threshold. People with 40 or more CAG repeats will develop Huntington's in their lifetime. People with 36 to 39 repeats may or may not develop the disease within an average lifespan. And those with 27 to 35 CAGs will not develop Huntington's themselves, but risk passing on the disease to the next generation. The Huntington gene carries instructions to make the Huntington protein. The DNA is used to create a messenger RNA that carries the code to the cellular factory that makes the protein. Too many CAG repeats result in a toxic form of the protein being produced, known as mutant Huntington protein. Mutant Huntington protein damages the normal functioning of neurons in many ways, including interfering with their transport networks and gene regulation. This causes them to malfunction and die prematurely, resulting in the symptoms of Huntington's. Huntington's is increasingly recognized as a disease of the whole brain and body. Areas of the brain responsible for cognition, personality, and movement are particularly affected, leading to the classic triad of symptoms in Huntington's disease. While research is ongoing, there is currently no treatment that can slow, stop, or reverse disease progression in Huntington's. The goals of current disease management focus on providing symptom relief, maximizing function, and optimizing quality of life. It's because of my family and Huntington's families around the world that I and other scientists continue to work to progress Huntington's disease research. It is a huge collaborative effort between researchers and family members that will bring about a new chapter in this fight against Huntington's disease.
All right, as you can see, small changes in the genetic code can have devastating consequences for an individual. But there also can be some genetic advantages of having these disorders or being carriers of this disorder. For example, it talks about in the highlight here, more than 1,000 years ago, there was a typhoid epidemic in Europe, but people that were heterozygous for cystic fibrosis had the ability to block the bacterium that caused typhoid fever, and so they were immune to the fever, and they survived. Those that weren't immune did not survive, and that is one reason the cystic fibrosis gene is more prevalent in people of European ancestry. Another advantage of one of these mutations or disorders with sickle cell anemia, those that have one copy of the sickle cell allele are generally healthy. They are not considered to have sickle cell anemia, but they are also highly resistant to the parasite that causes malaria. And as you can see in these diagrams, if you see, this would be a map of Africa, Europe, and Asia, and you can see where malaria is most common. And if you look here, you can see where the sickle cell allele is common, and if you notice, they overlap. So there is some correlation there. People that have sickle cell or have the copy are slightly resistant, and so that allele tends to get passed through the population because of that. There is an analyzing data section here that you can look at sometime on your own, and that will take you through some questions, and maybe you'll be able to understand the relationship between malaria and sickle cell a little better after looking at this analyzing data. If you would have any questions on the analyzing data, just send them to me through the class comments. Thank you. Moving on then, there are chromosomal disorders that are larger, where you sometimes you have a whole extra chromosome. You know, humans usually have 46 chromosomes, but what if you have 47 or 45, where you have an extra one or you're missing one? This is known as a non-disjunction disorder, and in the highlight there you can see non-disjunction means not coming apart. So how does this happen? Well, as you can see, sometimes during meiosis, the homologous chromosomes do not separate properly. And you can see that the red chromosomes here did separate properly during meiosis 1, but the yellow homologous chromosomes did not. And so they end up in one cell before meiosis 2, and then they separated again. And instead of having 4 cells, each with one red, one yellow, you have different numbers. Some cells do not have yellow. Some cells have 2. And so, excuse me, and what can this cause? Well, one thing it can cause is Down syndrome. Down syndrome we've talked about in class before. This is where you have more than one. You would have 3 number 21 chromosomes. You can see how this happens here. They're showing you the X chromosomes, but they're missing all the other chromosomes. But you can see you're going to have extra 21 chromosomes. And if that egg cell has an extra 21 chromosome and the sperm cell comes together during fertilization, you end up with a cell that has 3 21s instead of the normal 2, and that would cause Down syndrome. 2 other non-disjunction disorders would be Turner syndrome and Klinefelter syndrome. In Turner syndrome, you have a female that only has 1 X, not 2. And in Klinefelter syndrome, you have a male that has more than 1 X. Usually it's 2 X's and 1 Y. Notice you always have to have an X it seems that having the X is vital for survival. You can see how these disorders can be caused here. You can see the cell dividing, creating the cells during meiosis, and you have the cells on the left that might cause Klinefelter. You have this cell on the right that would cause Turner syndrome. So you get XXY on the left, which would be a male with Klinefelters, and you get just X with no X or no Y to pair with it, and that would cause Turner syndrome. Let's go ahead and check out another video here. I have one on Down syndrome that I think is pretty interesting, if I can find it. I was looking at it earlier. This one won't be too informational. It'll be more seeing how Down syndrome, or people with Down syndrome, can function in life and the joy they can bring to people. So, and it's about baseball, so you gotta like that.
He's the back of the tub man, and he is one in the front.
Teddy Kramer is fiercely loyal to his teams.
Is that you right there?
Yes, that's me.
Right there, right in the middle.
Yes, in the middle.
A lifelong Cincinnati sports fan, he was student coach and manager for the state champion Colerain High School football team in 2004.
That's a very exciting game.
Yeah.
It's fun.
Oh, if it's 50 to 10.
50-10 is a pretty good game.
Yeah, you need to relax a little.
Yes. Oh, yes.
A competitive swimmer himself, Teddy's got a room full of plaques, pictures, and medals documenting his accomplishments, recognizing his courage and spirit. Raised on Reds baseball as a kid, Teddy could rattle off lineups and statistics of his favorite players. These days, though, he has a much deeper connection to his hometown team.
Tell me what you like about Dusty.
Well, he like go way back.
Last summer, Teddy spent a night in uniform in the Reds dugout, about which the Cincinnati Enquirer wrote, Ted joined the Reds for a day, but changed the team forever.
He's a lot of fun. He came in the locker room before the game, and he's just like going up to everybody. He's like, my favorite players, yeah, I love your walkout song. And so he's always positive. So it kind of puts a positive spin on the dugout.
I kind of thought he'd go down in the dugout maybe and or go to the warning track and meet them. And, some of them would just, come over to him and talk, and then we'd go to seats and they'd say, this is the honorary bad boy. And we just never dreamed that it would turn into this kind of opportunity.
At a school fundraiser, Teddy's parents, Dave and Cheryl Kramer, won a silent auction to be an honorary bat boy with the Reds. The team's regular bat boy, Luke Stowe, showed Teddy the ropes.
When he came in, he was giving everyone high fives. I was introducing him to the guys, and he was real energetic and lively. And I knew right then that this kid, he's going to be something special.
Teddy's up. I mean, this guy's he loves being out there. He loves the spotlight. He loves the players.
Less than a year after that first night in the dugout, Teddy was back for a second stint, working between innings at Great American Ballpark. Third baseman Todd Frazier gave Teddy a wristband he'll always keep, then gave him a memory he'll never forget.
My last at bat before I got taken out, I'm standing on the step, and he snuck up behind me again. He's like, hey, come on, you can hit me a home run. Please hit me a home run. I said, all right, you got it.
That ball clubbed in a straightaway center field, hit a ton, and that one will fly out of here.
Lo and behold, I hit the home run, and ran to second, hitting third, and I see this big smile, and the guy he's pointing at me like this. And it was just a surreal moment because that means a lot, to me. To him, give him a big bear hug, and he's pointing to the sky, and it's like he hit that home run. It was pretty cool. He was with me right there.
He brings a lot of energy into the dugout, that's for sure. And of all the things the Reds have said about Teddy, it may have been said best on Twitter.
Brandon Phillips tweeted this. He said, Teddy came in here and blessed us with his energy and his presence. Enjoy life, be yourself, go out and play hard, give it all you've got. He's a reminder to us all. This picture and that tweet hang on the wall over his bed in Teddy's room.
He really connected with them on a level, and maybe they just saw that this was their childhood, when they were excited about being at a baseball game, and brought back kind of a joy and enthusiasm that they hadn't seen in a long while.
Nothing like this. I've not seen anything motivate and energize the clubhouse and the team like Ted has.
Hey Todd, I know you're watching at home. I'm gonna tell you this. The home run you made today, it's very cool. It's awesome hit, and I can't wait to see you again. I love you, Todd.
Hopefully, after watching that video, you have an understanding that people that have Down syndrome or other disorders, they're just not someone in society that we have to take care of, or that we have to watch after, and they cause problems for society and make our lives problematic. But they actually bring a lot of joy to society. They can bring a lot to people's lives, and we can learn a lot from them. So it's important that we as a society accept people that have these disorders, and then we make them a part of society because they do bring a lot. All right, guys, this kind of brings an end to our 14.2 lecture on human genetic disorders. Don't forget, you have lesson 2 assessment questions to look at. And if you would have any questions over them, just send them to the comment section of the assignment. It's been great talking to you today, not with you. I've just been talking to you. And hopefully soon we will be able to get together and have a normal class where you'll be able to interact with me, ask me questions, and I'll be able to answer them, and not just have a lecture like this. But this is interesting, it's different, and hopefully you got something out of it. See you soon.